what is sickle cell disease, or SCD? Sickle cell is a genetic blood disorder. This blood disorder is caused by mutations in the protein that carries oxygen in the red blood cells in your body, and that protein is hemoglobin. This mutation causes red blood cells to become rigid, sticky, break apart easier, and generally they get stuck in vessels and capillaries much easier than normal red blood cells do. In the U.S., approximately 100,000 people are affected by this, and it's millions worldwide. More prevalent in Middle Eastern, African, Indian, and Mediterranean ancestry. Sickle cells that block vessels and capillaries can cause severe pain episodes, chronic anemia, organ damage, and increased infection risks. So normal red blood cells are round, smooth, they kind of have an indent in the middle, and they flow really well through blood vessels and capillaries. And they're great at carrying oxygen because the hemoglobin is evenly distributed throughout the red blood cells. Now, sickle cells are more of a crescent-shaped, like a banana almost. They're very rigid, very sticky. They break apart easier because they're much more fragile than normal red blood cells are. And because of their weird, odd shape, they get stuck in blood vessels and capillaries more easy, which causes pain and organ damage in people that have sickle cell disease. Because they break apart faster, this can cause chronic anemia, meaning you're not making enough blood cells in your body because they're breaking down faster than you can build them. It can trigger inflammation, so like when it gets stuck in things, it causes a buildup of things and inflammation to rise in that area, which causes pain crisis. And it can cause fatigue and organ stress because you're not getting as much oxygen delivered because the hemoglobin in a sickle cell isn't distributed evenly, so they're not great at carrying oxygen throughout the body in red blood cells.
Normal hemoglobin, called HbA, has a specific amino acid, glutamic acid, at position 6 on the beta chain. This amino acid is hydrophilic, meaning it interacts well with water and helps hemoglobin stay dissolved in red blood cells. In sickle cell disease, a mutation replaces glutamic acid with valine. Valine is hydrophobic, so instead of interacting with water, it creates a sticky patch on the hemoglobin molecule. This causes hemoglobin molecules to stick together, especially when oxygen levels are low. They form long fibers that distort red blood cells into a sickle shape. These mishapen cells can block blood flow and break more easily, leading to the symptoms of the disease.
Identifying the mutation. Sickle cell disease is caused by a point mutation in the HBB gene on chromosome 11. This causes GCG to mutate into GTG in DNA, basically changing cytosine to thymine. After this, it gets transcribed into mRNA and then translated into an amino acid. We find that it originally should have been glutamic acid prior to the mutation, but after the mutation, it ends up being valine. This is the mutation that causes sickle cell disease. Glutamic acid is hydrophilic, which means it absorbs water and it's water friendly, but valine is hydrophobic, means it repels water and it doesn't like it at all. This causes the hemoglobin to become sticky in the valine, or because of the valine. You can be only affected by the sickle cell disease if both parents carry the gene and pass it down to you. So if one parent passes down the gene and the other one doesn't, then you're not affected, but you're still a carrier for it. In some rare cases, such as Sanaya's, even after both genes are passed down, she didn't show any symptoms of the disease. This was because her fetal hemoglobin didn't stop producing. Usually after birth, the fetal hemoglobin stops producing and the adult hemoglobin takes over, and this basically helps the red blood cells to become like sickle cells. Connecting mutation to symptoms. Since glutamic acid is replaced with valine, This paves the way for sickle cells to form. Valine causes abnormal hemoglobin S, and this causes hemoglobin to clump up together. There should normally be space between hemoglobin molecules, causing the blood cell to end up in a round shape. Since the hemoglobin is clumped together, this gives the cell a sickle shape. And in the image to the right, you can see that a normal hemoglobin molecules, they're spaced out and they make sort of a circular cell or circle shape of a cell. But the clumped up hemoglobin, it just comes together and it gives it that sickle cell shape and doesn't give it any space to expand. And these sickle cells, they have a hard time going around the body and often get stuck in blood vessels. And since they're so sticky, they usually stick to each other when they get stuck, causing the normal blood cells, the round ones, to not pass through either. And this basically gives the person that's affected with the sickle cell disease a hard time because they're going through a lot of pain when this happens. And it can be happening for hours to days at some points. And the round blood cells, when they get blocked off from going over there to those areas of the body end up getting damaged because the red blood cells couldn't get there and deliver their oxygen in time. And this in turn damages the body organs and tissues. And a normal red blood cell can live for around 120 days, while a sickled blood cell can only live up to 20 days at max.
We will look at the current and emerging treatments. Hydroxia increases fetal hemoglobin, HbF. This reduces sickling and pain episodes and also the need for less transfusions. Not all patients, however, will respond and require monitoring for lower blood counts. With blood transfusions, this provides healthy donor red blood cells, reducing the stroke risk and complications. It, however, can cause an iron overload and risk of immune reactions. For bone marrow transplants, this replaces defective stem cells with healthy cells. It is the only established cure and requires a matched donor. We will look at CRISPR gene editing. CRISPR edits a patient's own stem cells to target the BCL11A gene. This suppresses fetal hemoglobin. By turning off this gene, HbF is reactivated, preventing sickling and reducing pain crisis. It doesn't require donors, but it does involve chemotherapy, offering a promising, precise genetic solution.
Overall, sickle cell disease is caused by a single mutation in the HBB gene, which leads to abnormal hemoglobin called HbS. This causes red blood cells to become sickle-shaped, rigid, and sticky. These cells can block blood vessels and break down quickly, which leads to symptoms like pain, anemia, and organ damage. This shows how one small genetic change can have a major effect on the body. By understanding molecular genetics, scientists can better understand diseases and create targeted treatments, while current treatments help manage symptoms. Newer gene-based therapies like CRISPR are promising because they target the root cause of the disease.